Hello students, so today we are going to learn the fourth part of the chapter reproduction in lower and higher plants. In this part we are going to learn double fertilization, development of endosperm, development of embryo, seed and fruit development, apomixis and lastly parthenocarpy. Okay, now in the present animation slide you can see pollen grains coming towards the flower. It's attached to the stigma. Now from the stigma, as the pollen tube starts to arise, it passes through the style, reaches the ovary, enters the ovule and releases the male gametes. Now we learn the detailed part of double fertilization. Double fertilization is a complex fertilization mechanism in flowering plants, that is angiospermic plants. As we know that all the angiospermic plants are flowering. It was discovered by Navashin in the liliaceous plant like Lilium and Fritillaria. After a pollen grain has reached the surface of the stigma, it germinates and forms a pollen tube which penetrates the stigma, style, ovary chamber and then enters the ovule, as we saw in the first animation slide, where the pollen tube from passing through the style then reaching the ovary and enters into the ovule, then releasing the male gametes. The growth of the pollen tube is guided by chemicals secreted by the synergids. Now, the pollen tube cannot go on its own and reach a specific place. So, it is guided by the chemicals which is secreted by the synergids. It usually enters ovule through the micropyle. It is termed as porogamy. So generally the entry point is the micropyle, which has a small opening or pore, hence the name given as porogamy. Poro, the presence of small opening, gamy, fusion, hence the term given as porogamy. But in some plants or in certain cases it is found that to enter through chalaza, that term is called as chalazogamy. So certain times it passes through the chalaza. So you can see in the diagram below, C part, where the pollen tube enters through the chalaza, and in some plants by piercing through the integuments called mesogamy. Now sometimes it may pierce through the integuments, as you can see in this figure, B part. So it is mesogamy. Finally it penetrates embryo sac of the ovule through its micropylar, and finally through the micropylar end it enters into the ovule. So you can see here in the figure A, porogamy, where all the labeled parts, embryo sac, chalazal pole, antipodals, integuments, which is bitegmic. Egg apparatus consists of egg and synergids over here. Micropylar pole, the pollen tube, the polar nuclei, funiculus are the parts of it, and as you can see this red colored tube entering into the micropyle, the term porogamy. Second, B part through the integument, so mesogamy, and thirdly through the chalaza, so chalazogamy. Moving further, the pollen tube carrying male gametes penetrates in one of the synergids. So as it enters the ovule, it first penetrates the synergids. Watery contents of synergids are absorbed by pollen tube, which then ruptures and releases the contents, including the two non-motile gametes. So first it enters the synergids, burst open to release the two non-motile male gametes. As non-motile male gametes are carried through hollow pollen tube, it is known as siphonogamy that ensures fertilization to take place. So here another term. Now you came across the term porogamy, mesogamy and chalazogamy. Here the fourth term, siphonogamy. Why siphonogamy? The term siphon means tube. So as the male gamete are carried forward through the pollen tube, it is termed as siphonogamy. So remember all these terms. Further, syngamy and triple fusion are two events of sexual reproduction in angiospermic flowering plants. So two more terms over here, that is syngamy and triple fusion. Now syngamy, the term syn means fusion of the first male gamete and the female gamete or the egg cell, forming syngamy, whereas the second male gamete fuses with the secondary nucleus, forming triple fusion as two polar nuclei and one nuclei from the male gamete, forming three nuclei. So hence it is triple fusion. Further, syngamy is a fusion of haploid male gamete with haploid female gamete to produce a diploid zygote. So it forms a diploid zygote. Whereas in triple fusion, second haploid male gamete fuses with a diploid secondary nucleus, producing primary endosperm nucleus, that is PEN, P-E-N, that develops into the triploid endosperm. So another short type of question, what is PEN? It is primary endosperm nucleus, which is formed by fusion of the male gamete and the diploid secondary nucleus. So since there is a fusion of three nucleus, it is called as triploid fusion or triple fusion or triploid endosperm is formed. The zygote then further develops into embryo. Syngamy is a type of generative fertilization, whereas triple fusion is type of vegetative fertilization. Here both the male gametes participate, therefore it is described as double fertilization. So as both the male gametes are involved in the fusion, they are termed as double fertilization. So you can see here the label diagram from the textbook. It is micropyle, the small opening, synergist, ovary, male gamete, secondary nucleus, and triple fusion, that is which occurs in the polar nuclei along with the male gamete, the stigma, the pollen grain which comes and attaches onto the stigma, then the pollen tube formation, style through which it passes, ovule, syngamy, the egg and the male gamete, that is fusion takes place over here, and finally double fertilization occurs. Now how this pollen tube formation or how the movement takes place, in further you can see the animation slide of it. Now here as you can see the animation slide, how the process of double fertilization takes place. So first one, the flowering plant, which is angiosperms. Second one, you can see in the flowers stigma, style and the ovary, where you can see the ovule. In the third one, you can see the pollen tube carrying the two male gametes. So you can see very clearly the pollen grain on the stigma, the pollen tube forms and slowly moves through the style and carrying the two male gametes, which reaches the ovule and bursts open to release the male gametes. Okay, and then the fourth animation image, you can see how the male gamete which is released by the pollen tube over here, one which enters the synergist and bursts upon the two male gametes, reaches the egg cell, forming the first fusion, that is syngamy, and the second male gamete reaching the polar nuclei, forming the triploid fusion. Further, significance of double fertilization. 4 points over here. It is a unique feature of angiosperms. It ensures that the parent plant invests a seed with a food store only if the seed is fertilized. So if that is, if the egg is fertilized, then the formation of seed fruit takes place. The diploid zygote develops into an embryo, which further develops into a new plant. Further, once the fertilization occurs, it forms a zygote and the zygote further forms the embryo. Third point, the triploid pen, that is primary endosperm nucleus, develops into a nutritive endosperm tissue. It restores the diploid condition by fusion of male gamete, which is haploid, with the haploid female gamete through the process called syngamy. And also, fifth point here, it also helps to avoid polyembryony. So since there is a fusion of male and female gamete, the formation of zygote and further embryony. So here the polyembryony is avoided. Now you can see I added an image over here which clearly explains the starting point till the end point. So the flower having pistil, ovary, ovule, the stamen, anther, and you can see the microspore mother cell. Then by meiosis it divides into 4. Then further by mitosis. Out of the four, as we have seen earlier in the previous video, one of them which undergoes mitosis thrice, remaining three degenerates. So from 1 to 2, 2 to 4, 4 to 8. Then pollen grain, now here you can see the formation: 1 megaspore mother cell, 4 formation, 1 megaspore which further undergoes 3 mitoid division to form 2, 4, 8, which again rearranges themselves over here. 3 egg apparatus, that is egg cell and the synergies, 2 in the center secondary nuclei, and 3 at the chalazal end, that is antipodal cells. So you can see here pollen grain, tube cell, generative cell. Further, in case of male gametophyte, so male gametophyte, female gametophyte. Then in the embryo sac, once a pollen tube which is passed through the style reaches the ovule, bursts open to release both the male gametes, and the syngamy and triple fusion takes place. Further, formation of zygote, and then zygote, which is along with the triploid endosperm nucleus, further forms the embryo. The embryo which forms the seed. Now the seed along with the seed coat, and then further formation of young sporophyte which matures into a new individual or mature sporophyte. So you can see from the flower to the formation of a new plant. Now the further development of endosperm. So you have seen how the process takes place systematically. So with the help of the diagram or the figure, it becomes easier to understand. Now how the development of endosperm takes place actually, and there are three types. So the triploid primary endosperm nucleus repeatedly divides mitotically to form nutritive tissue called endosperm, the one which provides the nourishment. In post-fertilization changes within the ovule, the embryo and the endosperm are seen to develop simultaneously. So both embryo and endosperm grows hand in hand or simultaneously. The other cells of the embryo sac disorganize sooner or later, that is, they start to degenerate later on. The formation of triploid endosperm nucleus triggers cell division, which leads to formation of endosperm. So as we saw in the earlier animation image, where the diploid that is secondary nucleus along with the male gamete forms the triploid endosperm. Now here there are three types of endosperm which you are going to learn on the basis of mode of development. These are nuclear type, then cellular type, and the halobial type. Again, the image taken from the text, you can see here the oospore, the embryo, the endosperm. So that is in case of nuclear. In case of cellular type, again you can see the oospore, the cellular type, and the endosperm formation. And third, halobial type, again the oospore formation and formation of embryo. So we learn in detail about these three types. So starting with the first type, nuclear type. It is the most common type found in 161 angiospermic families. So it's almost seen in 161 angiospermic families. The primary endosperm nucleus repeatedly divides mitotically without wall formation to produce large number of free nuclei. So here it keeps on dividing mitotically, that is the primary endosperm, and there is no formation of wall. So there are more free nuclei which is produced in large quantity. Third point, a big central vacuole appears in the center of the cell, pushing the nuclei towards the periphery. So a central vacuole which is appears later in the center and pushes all the cells towards the sides, that is the periphery. Later, walls develop between the nuclei, hence multicellular endosperm is formed. So the wall formation takes place later on, that's why it is seen as a multicellular endosperm which is formed later. But in several cases, cell wall formation remains incomplete. For example, wheat, sunflower, and coconut, where it is usually incomplete. Now, in case of coconut, has a multicellular endosperm in the outer part and free nuclear as well as vacuolated endosperm in the center. So from the name coconut, you can easily understand the concept over here. Outer part multicellular endosperm and inner part free nuclear as well as vacuolated endosperm in the present. That's why you can find the liquid in the coconut in the inner region, where the inner part, which is the which can be consumed, and outermost part, thick fibrous part. Then B type, cellular. In some plants, division of triploid primary endospermic nucleus is immediately followed by wall formation, so that the endosperm is cellular right from the beginning. So they are cellular right from the beginning stage itself as they start to divide. It is mostly observed in 72 families in Dicot. Now here, unlike the nuclear type which is in angiosperm, that is in most of the species, here it is specifically in Dicot plant. Example, balsam, members in balsam family, petunia, edoxa, etc. Then, now these again the images of these edoxa, etc. put in the further steps so to understand how the plant looks like, what is the family of these dicot members like. Third, halobial type. It occurs in the order halobials of monocotyledons. In this type, or in this case, the first division of primary endosperm nucleus is followed by a transverse wall which divides the cell unequally. So here there is an unequal division. The smaller cell is called chalazal cell and larger cell is the micropylar cell. So here we can identify with the smaller being the chalazal cell and larger being the micropylar. Then the nuclei is in each divide by cellular or free nuclear division, and then walls develop between nuclei in the micropylar chamber, as we saw in the previous image, where nuclear how it appears, cellular how it appears, and halobial type how it appears as such. Lastly, it is intermediate between cellular and nuclear type of endosperm. So this particular type, that is halobial, is a mixture or middle type of nuclear and the cellular. So here example is Asphodelphus. Now here, as I mentioned, I put all the images, what examples we are just read out. Wheat, which all of you have come across in their daily life, that is we are consuming wheat a lot, right? So anatomy of the grain which is given. Now here, just to have a quick glance over the points, brush like border over here, brand, that is alluvial layer, hyaline layer, testa, that is seed coat, inner pericarp, outer pericarp, endosperm, germ, that is scutellum and the embryo and the hull part. Whereas sunflower, which again you might have observed, the seeds how it appears, that is of sunflower, inflorescence, ray floret, disc floret, and the corolla part, sepal, ovary, the bract, which is ray floret, and disc floret, which is bract, corolla, style, stigma, and the sepal and the ovary. Third, achene, that is a fruit of sunflower, which has locule, pericarp, seed coat, seed, and funiculus. And then petunia flower, you can see how it appears along with the seeds over here. Then the balsam, now balsam you might have seen very few in case of where it appears usually in the colder regions as such. Here you can see the different species of balsam: A. embellis, A. balsamia, A. faceri, A. grandis, A. lasiocarpa, and magnifica. So you can see how many different types are present or species are present, and how the shapes vary. Then a few more images of edoxa and Asphodelphus, and here some additional information, that is mosaic endosperm. What is this mosaic endosperm? Endosperm containing tissues of two different types, that is called as mosaic endosperm. So there are two different types present in the tissue. So in plant like corn, the endosperm contains patches of two different colors. It forms a mosaic pattern, as you can see in case of the corn kernel. So you can see how two coloration you can see. And if you just see the labeling part, seed coat in both the case, the endosperm, the embryo, this is a cross section of it, and the without the cross section. Now Coming to the next part, that is development of embryo. Now, the process of development of zygote into an embryo is called embryogenesis. As we know, after the fertilization, that is syngamy, fusion of male and female gamete, zygote is formed. Now, this zygote further forms the embryo. The embryo formation is called as embryogenesis. Next point, the embryo is developed at the micropylar end of the embryo sac. So you can identify the embryo which is present at the micropylar end. The growth of embryo triggers only after certain amount of endosperm is formed. After fertilization, the embryonic development begins. The zygote divides to form two-celled proembryo. So the zygote first divides to form a two-cell proembryo. As you saw earlier the image, how the zygote forms the embryo, later on seed formation takes place. Then larger cell towards a micropylar is called base cell or suspensor initial cell, and smaller cell towards the chalazal end is called the terminal or embryonal initial cell. Then the suspensor cell divides transversely in one plane to produce filamentous suspensor of 6 to 10 cells. Now, so you as we saw how the division takes place, even further you can see the image, that is the suspensor cell which is present towards the micropylar end, which divides into 6 to 10 cells. The first cell of the suspensor cell towards a micropylar end becomes swollen and functions as a haustorium. So remember the term, the suspensor cell, the first cell, they get swollen and forms haustorium. The lowermost cell of the suspensor cell is known as hypophysis. So the lowermost cell hypophysis, the first cell haustorium. The suspensor cell helps in pushing the embryo into the endosperm. The embryonal initial undergoes 3 successive mitotic divisions to form octane. Now coming to the second part, that is first the suspensor, now the embryonic part. The embryonic which divides by 3 mitotic division and further forms octane. As we know, 1 division to 2, 2 to 4, 4 to 8. The plane of divisions are at right angles to each other. The lower tier of 4 cells of the octane gives rise to hypocotyl and the radicle, whereas the 4 cells of the upper tier forms the plumule and the 1 or the 2 cotyledons. So as we see the formation of 8 cell stage, the 4 cells which forms the radicle and the hypocotyl, whereas the remaining 4 cells on the upper tier forms the plumule. As we know, this plumule forms the shoot and further the stem, and there may be 1 or 2 cotyledon depending on the type. Then the hypophysis by further division gives rise to part of radicle and root cap. Subsequently, the cells in the upper tier of octane divide in several planes so as to become heart-shaped, which then forms 2 lateral cotyledons and a terminal plumule. So here you can see 2 lateral cotyledons are formed and the terminal plumule is also formed, and this octane actually becomes a heart-shaped structure. Then further enlargement of hypocotyl and cotyledons result in a curvature of embryo and it appears horseshoe shaped, and further it takes the horseshoe shape. Now we can see this image again taken from the text. So whatever we learned now, we can see the different shape formation, how the changes taking place. So A, the oospore, B, the 2 cells proembryo. Then for there are various labeling mentioned, like B, the B is for 4 cell. Then I, the that is shaped embryo, proembryo part are formed. So different labels are given to identify. Then the C part, that is this part, it is the further development of embryo. Then in that case the S is the suspensor, the B part is the hypophysis, or you can say in fact it is a H which is the hypophysis. E, endo, that is embryonal mass. Okay, then further the D image is LS of ovule, where endosperm in a free nuclear stage. Then further the E part, embryo, showing further development of embryonic octane. So you can see here the octane stage which is formed, and you can see here the heart-shaped structure formation. Then F, LS of ovule of endosperm becoming cellular. Then G, embryo, that is cotyledons which are formed, in short COT, the cotyledons are formed, which again as we mentioned takes the horseshoe shaped structure. Then lastly the H part, that is mature seed, formation of the mature seed takes place, where you can see plumule, the endosperm has been consumed almost completely. So this is a development of dicot embryo as in capsula. Further points, that is in case of monocot. Now what we learned is in case of dicot. Now coming to the monocot. So the embryo development is similar in both dicot and monocot up to the octane stage. So the initial part is almost similar. The difference appears later. In monocot embryo, single cotyledon occupies terminal position and plumule in the lateral. The single shield-shaped cotyledon is called as scutellum, which again you can see over here. The protective sheath of the plumule is called coleoptile. So the protective covering is the coleoptile, whereas radicle is coleorhiza, which you can see over here, coleorhiza, and here coleoptile. So finally ovule is transformed into seed and the ovary into fruit. So here the formation of seed and fruit is completed. So just to go through the image, so here in figure 1.6, development of monocot embryo, A, you can see the oospore, and then B, which divides into two, as we had just learned in the beginning, the upper part which is the suspensor and the lower embryonal initial. C part, where you can see the division taking place. D, the division takes place further. E, we can see the suspensor cell, and here plumule and the cotyledon. F, the plumule, the larger part, the cells division takes place, suspensor, radicle, hypocotyl, plumule, and the cotyledon. So in G, we are very much clear with the plumule part, radicle, and the cotyledon. And finally H, scutellum, as we just read out, which is the shield shaped cotyledon, coleoptile, the sheath of the plumule, coleorhiza, which is of the radicle. Other than that, root cap, radicle, epiblast, and the shoot apex. Now seed and fruit development. So further again I added an image just to understand the seed and fruit development. So you can see here the monocotyledon with endosperm, cotyledon, coleoptile, epicotyl, hypocotyl, radicle, seed coat fused to ovary wall, which we just saw in the previous image as well. So here you can see the seed coat, the radicle formation along with hypocotyl, then the epicotyl on the surface of the along with the coleoptile over here, and the first leaf is formed. Whereas in case of dicotyledons, you can see very clearly two cotyledon, the epicotyl, hypocotyl, cotyledons. Then if you see how the progress takes place from the radicle to formation of the hypocotyl and the root, then the cotyledon, further the first leaf formation. The cotyledon also you can see here very clearly. Now in detail about the seed and fruit development. So here first we see the image first, so then it will be easier to understand the explanation part. Now in case of dicot bean seed, the epicotyl, hypocotyl, radicle, the seed coat. Okay, then here the two cotyledons and the entire embryo. Whereas in case of maize, monocot, seed coat and the fruit wall, alluron layer, the endosperm, this region, scutellum, coleoptile, plumule, radicle, and coleorhiza. So first point, the goal of reproduction in every living organism include plants is to create offspring for the next generation. That is, if the seed formation takes place, then the further next generation will be formed with the help of these seeds. Then one of the ways that plants can produce offspring is by forming seeds. That is very clear. The flowers must be pollinated in order to produce seeds and fruit. So first major thing is pollination. If pollination occurs, which will lead to fertilization. If fertilization occurs, then it will form the embryo development and further seed and fruit development. So seed development is initiated by fertilization. The integuments of the fertilized ovule persist and get transformed into seed coat of mature seed. Seeds sometimes consist of two distinct coverings, a typical outer coat which is testa and the inner thin membrane is that is tegmen. Whereas the nucellus in the ovule may persist in some genera like black pepper and beet as a thin papery layer which is called as perisperm. So here you can see the seed coat have a covering outer testa, inner tegmen. Also there may be a papery layer which is formed by the nucellus in certain species like black pepper and beet. In fact, the genus. In some seeds, the food reserves in the endosperm are partially used up in the development of embryo. Obviously, in such seeds, the endosperm remains conspicuous and fills a greater part of the seed. So, in seeds, the endosperm which remains in the seed itself so forms a greater region. Thus, the resultant seed is endospermic or albuminous. So if the seeds contains the endosperm, then it is endospermic or albuminous. The example, castor, coconut and maize. Further we can see all the images as well. Now further point, so first we'll go through the point, then the images. In other seeds, embryo absorbs food reserve from the endosperm completely during its developmental stage. Thus, the endosperm disappears in the mature seeds. Now, in certain case, what happens? The endosperm is used up as the food. So the endosperm does not have, the matured seed will not have the endosperm. So then the resultant seed will be what? Non-endospermic or X-albuminous. That is in for example pea or bean, etc. The cotyledons in some endospermic seeds act as a food storage and in others they are the first photosynthetic organs. Micropyle persists as a small pore in seed coat to allow the entry of water and oxygen during soaking. So here if I give a small example, at home when mother keeps the seed overnight soaked in water, what happens? The next day it swells up. Also we can see the formation of the sprouts. So we can see sprouting occurs. So this takes place through a small pore present in the micropyle. Further, the fruit development is triggered by the hormones produced by developing seeds. So when the seeds develop, it is triggered by the hormones. After fertilization, the zygote is formed and the ovary begins to differentiate into fruit and the ovary wall into the pericarp. So the pericarp is basically three-layered, which gets differentiated into fleshy fruit like mango, coconut, etc. So just to make you understand much more clearly, I added two more images, one of germinated coconut. Now again just a little bit of additional information over here. If you take the coconut, there may be three holes in it, but or darkened region in which one hole will be functional where you can pierce through it, other two holes will be very hard which is actually plugged pore and one functional whereas other two are usually plugged. Okay, and then over here we can see the root, mesocarp which is a fibrous part, shoot, the stem and leaves, the coconut apple that is the inner part which we can consume, that is usually a spoon, that is a spongy, a sweet mass, cotyledon which is that mass dissolves and absorbs the endosperm, and the coconut meat endosperm. Exocarp, the outer layer, mesocarp, fibrous husk, and the endocarp, which surrounds the seed, which is the hard part of it. Then in case of castor seed, kernel after removal of seed coat. So here you can see endosperm, testa, tegmen, cotyledons, embryo, and here the caruncle, the hilum and the seed coat. So whole seed cut into half showing position of the embryo. So here is a whole seed, and when you make this section you can see how it appears inside, and here the kernel part. Now few more images. So I always add images which helps you to understand the concept much more clearly. So here also you can see pea seed. Okay, the embryo, the testa, seed coat, plumule, hypocotyl, radicle, The embryonic axis, the storage of cotyledon. The pea plant, otherwise Pisum sativum, which we have learned in the first chapter, which was the study carried out by Mendel, right? Then parts of the fruit, mango, otherwise Mangifera indica. Why indica? Species made in India, Indian product or Indian species, right? Okay, further, epicarp is outer skin, middle mesocarp, the fleshy which we consume, the edible part, and the inner endocarp, the stony wall, together forms the pericarp. And the embryo, that is the inner seed part, we can say, baby plant, endosperm which is a reserve food, and the seed coat together forms the seed. Now, what is the significance of seed and fruit formation? The importance. What is the importance? Fruits, which provide nourishment to the developing seeds. Fruits protect the seeds in immature condition. Third, seeds serve as important propagating organs of plant. Means it helps to further produce new offspring, right? Seeds and fruits develop special devices for their dispersal and thus helps in the distribution of species. So when you say special devices, it may be with appearance to the fruit, the texture, the shape, size, all these things. So every plant, if you see, the fruits are of different shape, size, color. So that is different, which acts as a devices for them for dispersal. Just to add one more point over here, success of seed as a propagule is due to many characteristics of seeds which are as follows. Dormancy, right? We have learned these terms last year. Dormancy, that condition where the seeds remains dormant. Viability, how they can mature even after a certain period of time. Reserve food which is stored in it. Protective coat, that is a testa tegment. Dispersal, how they are dispersed by various ways, maybe wind, water, animals, etc. Then edible fruits which are consumed. Right. Now, just to explain here, what is dormancy actually? First point, dormancy is a state of metabolic arrest that facilitates the survival of organism during adverse environmental condition. Structural or physiological adaptive mechanism for survival is called dormancy. So, the adaptive mechanism for survival is called dormancy. Mature and viable seeds will not germinate even in the presence of favorable condition, and they are dispersed at different places during dormancy. So, they can be dispersed and can be stored for a temporary period of time. As if I give a simple example, at home, as mother stores the seeds in a container and whenever required can be kept for sprouting or for cooking purpose, right? Or even we can germinate those seeds. Viable seeds germinate only after completion of dormancy period. Now again few more images, all beautiful images you can see. First read the caption which is given in the textbook, also additional information. How long seeds stay viable and healthy, means the time period, maybe 1 day, 1 week, 1 month or 1 year or more than that. Can old seeds still grow? Is it possible? Some examples of oldest mature seeds that have grown in viable plants are as follows. So these examples are given in the textbook. That is Lupinus arcticus, which is 10,000 years old. You can imagine 10,000 years. That is why I put the images over here. See, you can beautiful flower of Lupinus arcticus along with the seeds, which is 10,000 years old. You can imagine how old. Even we may not be there because it's 10,000 years. We can say 100 or 1,000 times our age, right? Then Phoenix dactylifera, 2,000 years old. Again, I put the image Phoenix dactylifera, or in simple words, as you know, date palm, right? Which is almost 2,000 year old. Some seeds are short-lived. Example, citrus. Now citrus, the lemon or any citrus fruits, if you take, the seeds are short-lived in case of citrus. Some tiny seeds are easy for dispersal. Which one? Strigo, orchids and Orchibranca. So striga, you can see again I put flower, again very beautiful flowers, and the seeds are very minute. That's why the dispersal is very easier. Orchids, again there are very beautiful flowers of orchids, right, which the seeds are again very light. You can see here in the hand small hair like structure or finer than a hair which is present in the fingers, many numbers, so which can be easily dispersed. Then orobrancha, see how the shape of the flower itself is, or plant also, and you can see the seed is just 0.5 mm, so minute it is, it will be not even visible at times, right? 0.5 mm is very very minute, so small it is. So these plants like striga, orchids and orchobrancho, the seeds are so minute it can be easily dispersed and spread out. Now, coming to the next part, that is apomixis. Now, apomixis, it is a phenomenon of formation of embryo through asexual method of reproduction without formation of gamete and the act of fertilization. So, up till now in the previous three part of the video and present topic, we are learning about reproduction. So, now we are learning apomixis, where the formation of embryo takes place asexual method. So, without undergoing fertilization or any fusion of gametes or formation of gametes. So alternative is unusual sexual reproduction, where there is no meiosis and syngamy. As I told, no fusion. Embryo develops into ovule and ovule develops to form seed. In apomixis, when a gametophytic organ or cell produces embryo-like structure without fertilization, it is called as apogamy. So again, these are all one word terms: apogamy, apospory, apomixis. So I repeat again, what is apogamy? In apomixis, when gametophytic organ or cell produces embryo-like structure without fertilization. Now similarly, when diploid sporophyte cell produces a diploid gametophyte without undergoing meiosis, it is called apospory. So here it is the sporophytic cell. In case of apogamy, it is a gametophytic cell. That's why it is apogamy. Here it is apospory. Example, orange and mango. So just to make it clear, I put a chart image of type of apomixis: gametophytic, sporophytic. Gametophytic, we can see here unreduced cell gives rise to a megagametophyte. The embryo sac originates from the megaspore mother cell either directly by mitosis and or after interrupted meiosis. Then apospory, the embryo sac originates from nucellular cells, the most common mechanism of apomixis in higher plants, characterized by the presence of multiple embryo sacs or androgenesis. Then in case of sporophytic, unreduced cells gives rise directly to an embryo. So adventitious embryony, parthenogenesis and apogamy. So what is, and one more term is given, semigamy. The haploid sperm nucleus enters the egg but does not fuse with the haploid egg nucleus. Each nucleus divides independently, creating a haploid embryo that contains sectors of male and female origin. Now we can go into the types. The main categories of apomixis are recurrent apomixis. In this type, the embryo sac generally rise either from a archisporial cell or from some other part of the nucellus. In diplospory, the unreduced embryo sac is derived from the diploid megaspore mother cells. From the diploid MMC, that it is derived. Hence, it is diplospory. Example, Taraxa cum. In apospory, the nucellular cells gives rise to apomictic embryo sac. So that is in case of recurrent apomixis. If it is non-current apomixis, the second type, in this type, the megaspore mother cell undergoes unusual meiotic division, not the usual one, that is undergoes usual meiotic division, which is other than what is seen, and a haploid embryo sac is formed. Here, embryo arises either from the egg by parthenogenesis or from some other haploid cells of gametophyte through apogamy. That is, doing the with the help of the gametes, or here as you say gametophyte. Plants produced by this method are generally sterile, so they are not fertile but sterile, so they cannot produce further and do not reproduce sexually. Example, Nicotiana. So when you say Nicotiana, if I add the species over here, it is Nicotiana tabacum, the tobacco plant. C. Adventive embryony. In this type, embryos may develop from somatic nucleus. When is somatic? The body cells, as you learn. So nucleus of the cells, or integuments along with normal zygote embryo. It is common in mango, orange, lemon, etc. So this embryony is more commonly seen in the citrus fruits. It gives rise to conditions called polyembryony. Okay, that's why we can see many seeds in orange, lemon, etc. That's why polyembryony is formed. Genetically identical plants can be produced effectively. So here again I put another chart which gives the definition in short with their types. So type of apomixis is based on occurrence. Non-recurrent embryo develops from any cell of embryo sac. That is haploid. Haploid parthenogenesis, embryo develop from the egg cell. Haploid apogamy or pseudogamy, which embryo develop from synergids or antipodal cells. Recurrent apomixis, that is embryo sac develop without meiosis from the diploid cell. Diplospory, embryo sac develop from MMC, that is megaspore mother cell. Apospory, embryo cell develop from any vegetative cell of ovule. Then embryo is developed from the diploid egg cell. Third type, adventitious embryony, as we learned, it is sporophytic budding. Embryo directly developed from nucellus or integuments. No production of embryo sac. Example, mango, citrus. So it is just a short form of what you have learned. Now the images, that is taxacum or dandelion. Again you can see the flower, how beautiful they are. Then nicotinia. Here mandarin, that is citrus reticulata, or you can say the citrus fruits. Then here polyembryony, how it occurs. So you can see a zygotic embryo, endosperm, and the antipodal embryo, or this end antipodal end, so polyembryony. Then the later part of the chapter, that is Parthenocarpy. This is a term coined by Knoll in 1902. It is a condition in which fruit is developed without the process of fertilization. It occurs naturally in varieties of pineapple, banana, papaya. So here what is Parthenocarpy? That is, the fruit is developed without undergoing fertilization. That is why in pineapple or banana you don't find seeds. In these plants it seems that placental tissue in the unfertilized ovary produces auxin IAA indole 3 acetic acid, which is responsible for enlargement of ovary in the fruit. That's why the fruit is enlarged or larger in size. The fruit resembles a normally produced fruit, but it is seedless. As I mentioned, in banana, pineapple, the seeds are absent. So you can see here natural parthenocarpic, the banana, pineapple, or even in case of watermelon also it can be carried out. Here parthenocarpy, you can see the fruit, or that is in can take the example here tomato, where the seeds are not seen, or here it is mangosteen, seeds are seen, but by parthenocarpy you can see pseudo embryo, very minute seed like structure, which is a pseudo, means false seeds. Then do you know parthenogenesis is the development of embryo directly from the egg cell or a male gametophyte. It is a kind of apogamy. So agamospermy, here the plants produce seeds, but embryo inside it is produced without meiosis and syngamy. Parthenocarpy can be induced artificially by spraying of gibberellin, delaying pollination, use of foreign pollens. That's why in watermelon seedless it can be carried out artificially. Genetically uniform parental type seedlings are obtained from the nucellular embryos. Now further, the polyembryony. Now we have come across parthenocarpy. Now the polyembryony, we have come across a term in between the earlier parts. So in detail about it. So it is a development of more than one embryo inside the seed, and the condition is described as polyembryony. The term poly means many. So formation of many embryos. It was first noticed by Leeuwenhoek. Now all of you know Leeuwenhoek, Anton von Leeuwenhoek, the scientist who found the first microscope in the seeds of citrus genus. It is the occurrence of more than one embryo in a seed, which consequently results in the emergence of multiple seedlings. So many seeds are formed. The additional embryo resulting from the differentiation and development of various maternal and zygotic tissue associated with the ovule of seed. Polyembryony may be true or false depending upon whether many embryo arise in the same embryonic sac or in case of different embryo sacs in the same ovule. So it may be true or false. In adventive polyembryony, an embryo develops directly from the diploid cell of the nucellus and integuments as in citrus. In cleavage embryony, the second type of polyembryony, zygote proembryo sometimes divides into many parts or units. Each unit then develops into an embryo. Polyembryony increases the chances of survival of new plants. So it helps to increase the survival as many embryos are present. Lastly, nucellar adventive polyembryony is of great significance in horticulture. So here just an image showing the development of embryo, and secondly adventative polyembryony, that is you can see A, B and C part. So BC is both adventative polyembryony, and here you can see how polyembryony appears and monoembryony looks like. Mono means one single embryo, poly many embryos. You can see 3 embryos over here. Then again in short, the introduction of polyembryony we can learn in short. That is, polyembryony is a phenomenon of 2 or more embryos developing from a single fertilized egg. That is, in case of humans, what it is, identical twins. Polyembryony occurs regularly in many plants and animals. Example, parasitoid insects, where it can be observed. Discovered by Leeuwenhoek in 1971 in Citrus aurantium, coined by Brown. So the term was coined by Brown in 1859. It is main observed in gymnosperms than in angiosperms. So moreover seen in gymnosperms. So polyembryony observed by Anton von Leeuwenhoek in 1719 citrus fruits. So you can see here how it takes place here inside. Usually we have 8 cell stage. Here we can see synergids and the more filiform apparatus also seen over here. Then reasons for polyembryony can be, it is due to the formation of more than one egg in a embryo sac. The formation of more than one embryo sac due to integument and nucellus cell. Synergid cell, antipodal cell may also develops into embryo. So synergid antipodal can also get converted into embryo. That's how one of the reasons of polyembryony. So orange, lemon, groundnut, mango, onion, etc. So you can see here zygotic embryo, that is as just we learned in case of synergids or in case of antipodal cells, even they can be converted into embryo. Then just few more additional information. Ernst and Scharnoff classified the polyembryony two types: true polyembryony, false polyembryony. True polyembryony can be further divided into two types: cleavage polyembryony, adventive embryony. So what is the importance? Plant breeding, horticulture, nucellar embryos are supposed to be free from disease. Propagation of fruit free such as citrus and mango. Application of adventive embryos is also important for providing genetically uniform seedlings in fruit trees. Can be used for development of homozygous, diploid, artificial production of these embryos from the eggs or synergids. So these are the important. Okay, so that is the last part and the assignment question based on today's part. So by this we end the chapter. So thank you all of you.